Welcome to our webinar on the Nexus of Climate Change and Malaria. Welcome to our webinar on the Nexus of Climate Change and Malaria. Thank you everyone for joining us today. This is our first webinar series hosted by Environmental Economy Unit at HITAP. My name is Yimeton. I'm a research assistant in this unit, and I'll be your moderator for today's section.
Good afternoon, everyone. I'm Pantira Sapankeo. I am research assistant for this unit from HITAP. Today, I will be your moderator as well. And before we begin, I would like to go through a few housekeeping notes for today's webinar. Okay, for today's housekeeping note webinar, first, the webinar will be recorded and shared on our YouTube channel. Second, please keep your microphone muted during this session. Third, feel free to use the chat box to ask questions or share your thoughts during the webinar. And we will have the Q&A section after the presentation of speaker. You can raise your hand using the Zoom feature if you would like to ask questions directly.
Okay, now I'm going to introduce our special speaker. We have research professor Jetsumon Prachumsri. She's the head of Mahidol Virus Research Unit, Mahidol University. She has over 30 years of experiences on malaria research and working with endemic population in Asia. She is also well known for malaria transmission study, especially on Plasmodium vivax. And she's also a key opinion leader on malaria elimination programs. And then we have Associate Professor Kimberly Fornace from National University of Singapore, London School of Hygiene and Tropical Medicine. She's leading the Climate, Environment, and Health program at NUS, and she's well known for her research in spatial epidemiology, especially in understanding the impact of environmental changes on infectious disease dynamic. She also experts on using technology, especially on satellite data and machine learning, which made a big impact on malaria research and control strategy in Southeast Asia. So now that we know our speaker, I will move on to the agenda item for today. So this is the agenda for today. We will have two speakers. They will talk around like 20 minutes each. So I think Jetsumon will talk for the first person to deliver the presentation. She will talk about climate change and malaria in Thailand, and then followed by Professor Kimberly. She will talk about her observation data to monitor climate and malaria risks. And after the presentations, we will have panel discussion for 25 minutes. And this is where the expert will answer all your questions during the registration. And also, if you have any question during the webinar, feel free to put it in the chat box so the expert can answer your question. And then we will end our webinar with the summary and closing by me and my colleague, Pantira.
So at this moment, I would like to invite Research Professor Dr. Jetsumon Sattapmongkot Prachumsri to speak on the first topic, please.
Thank you very much. I hope everyone hear me clearly. It's my honor to join the group today. I hope that I learn something new from my co-team, Dr. Kimberly as well. And then I would like to start with some simple information about malaria in Thailand and what we think about the climate that may impact in the area of my study. So you can hear more fancy stuff about the climate change from Kim later. So I start first. So my presentation today will cover about the climate change and what we think is a challenge to malaria transmission, the link between the climate change and malaria, and what happened in Thailand, especially at my study site, and what we think that it can be action plan to deal with this change and the transmission. Oops, doesn't move. Okay, briefly, oops, now it moved too fast. Let me go back. Yes. The climate change, what the climate change or anything that change from what we experience, when I was young, because I'm a little bit old, so I can say that the climate in Thailand changed a lot. We used to have a very long winter, that the real winter. Most of the Thai people now say that Thailand has only hotter and hottest, no more winter, right? So, but what happened with the climate change, right? We know that the sea level is higher, the land sink, rainfall pattern change. We have a lot of heavy rain and some drought and some long delay in dry season in certain area. And what happened, the deforestation, so we're not so sure whether the climate change, what the cause, what the impact from that. And of course, a lot of things that happen because of the man-made with we all need to be concerned, and then we can see whether we can make it better. Uh, this one, maybe next slide. Lily, can you share for me? It'll be faster. So, like I said, the problem, especially if you are living in Bangkok, you know that we experience flooding. In Thailand, we have heavy flood that never happened before in the northern part of Thailand. Okay, and so many things happen because we have less tree, you know, and now a lot of people try to make building or planting more tree to help. I'm not so sure whether it's enough to change things that already move forward in a bad way. Next one. Okay, so this is the trend of malaria transmission in Thailand for the past 10 years. Okay, you can see that the country also commit to malaria elimination by 2030. And we have the resurging of the malaria after 2021. In fact, during the COVID lockdown, we still see some rising already. So that alarm all the people that have to take care of malaria management in Thailand to think about what can be the cause. And the problem of malaria in Thailand that left for now, normally along the Thai-Myanmar border. Okay, we don't know whether the climate change also contribute to this. Next one. Yep. So in malaria transmission, a lot of people always think that climate change will impact a vector-borne. If the global warming come up, the vector-borne will be increased. But is it true in case of malaria? Malaria is a complicated disease. You need parasite, you need a vector, and you need the human host at the same proximity that the disease can maintain the transmission. So individual factor that contribute to the transmission have different things that can impact them. Let's move to the next slide, please. You see, so whether the climate change can be one factor, it will depend on how the climate change impact on those three things. Move to the next slide, please. Yep. So when the climate change, if the temperature increase, in some area, for example, like the Korea, Korean is in temperate zone, right? They have a lot of Vivax and long incubation Vivax. And the reason that the transmission happen only 6 months a year, because that's the only time that the weather warm enough that the mosquito can fly. But let's imagine if the climate change, the weather warmer, the month that you can have mosquito is more than 6 months, that means the transmission can be longer. So the disease can be increased. Korea hasn't successfully eliminated Vivax yet, and still not so sure whether they can do by 2030. The humidity will impact on the mosquito breeding place, but too much water or flooding also eliminate the breeding place. So it have to be proper ratio of the water source, that the vector can survive and maintain their life cycle. So in terms of host, human or animal, see when the climate change, how that impact for their agriculture or their living around there. So that will force the movement of the people from one area to another area and maybe increase the risk or decrease the risk, depend on where they move to. Therefore, some of the temperature will also impact their behavior on protecting themselves. For example, in Thailand and many country in Asia, the vector not very abundant, not like a vector in Africa that when you sleep outside, there are hundreds of mosquitoes come to you. But in Thailand, not many in most of the season, maybe less than 10 by you per night, and you didn't feel it. And if the weather is so warm, you don't want to stay under the bed net or in the house. So that give you more chance to get infected if you live in the area that already have transmission. I'm not very good at this. Okay. I would touch a little bit about monkey malaria, which Kim going to explain more on this. But recently in Thailand, we also face increased the case of penolicide, especially in southern Thailand, as you can see in the graph. But in fact, it starts from southern border, and we always blame that the movement of nosai is imported case from Malaysia. But Kimberly can correct me because after start the project, we found that Malaysia also said that the southern part of Thailand is also the source of penoxide to that peninsula Malaysia. Okay, they don't blame Sabah Sarawak move to that because it's an island, but I'm not so sure whether it's true. We will explore about that later. So human behavior, like I said, the weather in Asia, most of the area are hot and humid. So if you check, no one, this is the housing that show about majority of the house construction in many malaria endemic area in Thailand, especially in Tha Song Yang district, where is the our study site. The house look like this, to protect from the mosquito, you have to use a bed net, but when the weather is so warm, so humid, you don't want to stay under that. It's not very comfortable. The bed net that distributed by the government, after we did the study to check who use it, less than 60% of the population will use it. And one thing that the reason that they don't want to use it because it's uncomfortable. Okay, so then how the climate change impact the vector? Like I said, the mosquito depend on the physical factor, the humidity, the light, the temperature. So if the temperature, the humidity, or the light change in the way they're not favorite, then the vector will be declined, the vector population will be declined. But if the weather, the humidity like humidity is higher, you see the breeding place can be more than before. So that will have potential to increase the vector ecology and then the breeding site and more mosquito. But in general, with the high temperature that increase the deforesting, the land use change, generally you also change the species composition of the vector. And in many areas, the vector decline, not increase in terms of the population. This is study done by Kotbui. This one is very nice. I found that this project evaluate the environmental change and map that with the malaria cases. And you can see that the red map is about temperature. So the different colors show that this study follow for 5 years. So they map the temperature on each area in Thailand that found malaria and find the correlation whether you see the very dark red, if you can differentiate how dark of the red, that means that every year this temperature related to malaria cases. Okay, they have 1 year related, 2 year, 3 year, 4 year, very nice. The red one is the temperature. The humidity is the purple one. So the dark purple is mean that the area that have strong correlation between humidity and the malaria cases. And the green one is a rainfall. in general, if you map all spot together, you can see that the very hot spot is along Thai-Myanmar border, which the temperature, the rainfall, and the humidity is together that promote the breeding place and the transmission of the disease. This is another paper study in Tha province about the temperature. The top green one, okay, let me see that. The top green one is the average daily humidity. Not much different, right? The case decline. So even you see that this is contribute to malaria transmission, but not the direct impact of the malaria transmission. This one hard to say because even you see some correlation, but the total data that really contribute is very low. It's only less than I is only 0.39. So I would say that not much direct impact in terms of how the climate change impact on the malaria transmission. Okay, besides directly to the vector. This one is a study, is a data that we track from the database of the MOH. And as you can see that we see more increasing from 2020 to 2024 in that province, but the data of the temperature and climate not the change that much. So in this area, I would say that have less impact. So this picture show the study site. Okay, the upper one is one village in Tha Song Yang that the surrounding is more forest type compared to the bottom one, which is more like less forest village. We did the vector survey in these two study early this year, okay, to try to see the species composition because the ecology, the environment a little bit different. And we know that in Thailand and many areas in Asia and Southeast Asia that the species, the anopheles species composition is huge. You know, in one area you have what you know as a major vector and so many secondary vector that we want to understand how this impact on malaria transmission in these two sites. The way we collect the vector, we use CDC light trap compared with human landing catch and also larval sampling. So this is just the first part of the collection that we just did last year as a beginning of this third round epidemiology study of our malaria project. As you can see, the composition in the area, even the village is not that big. You see many species. Okay. We also collect some of blood fed by the light trap and as well as the human landing. And the majority of the vector, the highest number is always the by human landing is minimus group. Okay, as you can see. But the interesting thing is that we also found more Barbarostrix now, which we never seen that high number before. And this also similar as one study site in the southern Thailand, which I did not show in this study. So the biting rate and the biting time, okay, this is something that from human landing, so we also compare in two villages. You see the red and the blue one. The thing is that even is the minimus, okay, you can see that it's slightly different. It depends on, we believe that it depends on the activity of the villagers. That's slightly different, but we haven't collected the detailed data yet. So for larval density, okay, it's go along very well with where we get more adult. Normally, we also get more breeding place and more larvae from the sampling. And so this is just summarize what we found for the larvae instead in different area and in different breeding place. Okay. This one normally we found the small pool around the housing that we select to collect the vector. And in a more intermediate shade level, see like 50-70% in high forest village. That's the area that we found more adult. So we think that this is where the mosquito like the most. That's why you get more larvae, and the identification of the species is underway. We haven't finished the first laboratory analysis yet. So there's some action plan that we would suggest if you work on vector biology, and if you want to get a better plan, a better strategy to do the vector control in this area, like the vector surveillance. Okay, I found that it's hard. I always admire all of those entomologists that try to do hard job in terms of survey of the vector. Unless you identify the right vector, it's very difficult to evaluate whether your vector intervention or the vector measure is good enough. And without the good vector control, it will be very difficult to move forward for malaria elimination. You can control, but you cannot eliminate. Okay. And that's it. Okay, this is the team, and my study. We have been doing malaria survey in this area over 14 years now. Okay, with different grant support by NIH, and I hope that we still get continue the support. Okay, let's see, from the NIH, US NIH. Thank you very much.
Thank you very much, Professor Jetsumon. That was an interesting and insightful presentation. So for the question for this topic, we can keep it for panel discussion session. So shall we move to second session for today's webinar? So now I would like to invite Professor Kimberly Fornace to speak for second topic, please.
Right. Well, thanks very much for inviting me to speak today. And it's a great honor to speak after such a well-known malaria expert. So I'll speak a little bit about some of the ways we've been thinking about how we can use data to actually measure some of the environment and climate impacts on malaria within Southeast Asia. So one of the things that we've been very interested in is that we know that there are these strong relationships between different types of environmental variables and the malaria risks that we face. We also know that there's a lot of sources of data that can be used to actually collect and understand some of the climatic factors and the other environmental factors that impact the distribution of malaria. So we've been thinking a lot about how we can combine these two sources of data. So often this environmental data is collected for a different purpose by different agencies, but there is an opportunity to think about how do we use this data? How can we process it into the information or the variables that we need? How can we combine this with different types of epidemiological data on malaria or entomological surveillance data to really understand what the impacts of these changes are on infectious diseases, and ideally use this to actually mitigate some of these impacts. So just to kind of give one of the examples, looking at kind of wider climate patterns, this is work that was done in the Philippines with the Research Institute for Tropical Medicine, but looking at this island of Palawan, which reports most of the malaria cases for the entire country, where the cases are very concentrated in this southern area. So we have been working with RITM for quite a bit of time to think about how can we understand what are some of the factors that are driving these risks. So aggregating different things like temperature, rainfall, elevation, and other kind of processed layers such as population density to understand how these different drivers interact to drive malaria incidence within this region. So just looking at this visually, I think you can see if you look at a time series of this data, there's very clear seasonal patterns of malaria. So we see that every year there's kind of a peak and a decrease. So there is a very strong seasonality. But we do see particular years where we have very, very high numbers of cases relative to the other years. And some of the work that's being done by our group at NUS with RTM is looking at actually how can we combine some of this really kind of high resolution malaria case data with different indices related to environmental factors. So looking at this one highlighted here of the Palmer drought severity index, this is actually these shaded periods indicate times which are droughts, so have decreased rainfall or less rainfall than usual. And when we look at these are also correspond to some of the years where we have the highest cases. So just some preliminary work looking at specifically the response between drought and the malaria incidence. Of course, when there's a drought, so there's no rainfall, we also don't find that there's many malaria cases. But if we look kind of across time, so a period of time after this drought is reported, in this case, several months after the drought, there do tend to be higher malaria risks. So there's some very complex patterns between some of these climatic factors and malaria risks. And I think there's still a lot of work to do to think about what are the impacts of climate change and the changing patterns of seasonality on how we would expect these risks in the future. And of course, while we look at wider climate changes, this is, as Jetsumon has already said, this is not just about climate change, but this is also about other environmental changes. And one of the ones that we're really particularly concerned about is land use change. So specifically looking at deforestation and reforestation, which has been one of the major drivers of land use change across Southeast Asia. And I think one of the examples that we have where we see one of the most pronounced effects of land use change on malaria risks is actually looking at the emergence of zoonotic malaria. And so I think this was already touched on briefly, but for anyone not familiar with this parasite, Plasmodium knowlesi is a parasite which is typically carried by macaques and other non-human primates and transmitted by Anopheles mosquitoes. So a person become infected after a mosquito bites an infected macaque and then goes on to bite a person. But really in Malaysia is where we've seen the strongest evidence come out that this is such an incredibly important public health issue within this region. So this graph here actually shows the number of malaria cases reported from 2000 to 2018 in Malaysia. What you can see is there's been a hugely effective malaria control program at reducing some of the non-zoonotic human malarias. So there's now no indigenous cases of falciparum or Vivax or other human malarias. But during the same time, and particularly over the kind of past 2 decades, we've seen this increase in the number of human cases of Plasmodium knowlesi. So that at this point in time, Plasmodium knowlesi is the only indigenous cause of human malaria in Malaysia, and this causes thousands of cases in people every year. And this is, of course, important for public health because of the malaria burden, but also this can cause very severe and fatal malaria infections. So when we look at this graph where we see this kind of very clear trend and these increases in cases, one of the first questions I think that comes up is why? So what are some of the reasons we're starting to see this increasing trend? And certainly part of it is because of the diagnostics. So of course, once you're aware something's a problem and you start looking for it, you tend to find more and more of it. But even adjusting for some of the diagnostic uncertainty, there's been a very clear and strong relationship between human Plasmodium knowlesi risk and land cover change. So some of the earlier studies that were done in Sabah and Sarawak identified that the clinical cases of Plasmodium knowlesi were actually coming from areas where they reported some type of forest exposure, so movements into forest environments. Later, when we conducted a case control study in Sabah, we looked at clinical Plasmodium knowlesi cases and people who were living in the same villages who didn't report the disease. And we identified that people who had different types of forest and plantation activities had an increased risk of Plasmodium knowlesi at an individual level. And kind of finally, we've done much wider studies to look at population levels. What are the role of some of these factors? And we've seen that factors like deforestation and particularly forest fragmentation is associated with increased risks of exposure and infection to Plasmodium knowlesi. And so I think this kind of comes back to what Jetsumon presented earlier, in that we do see the impact of these environmental changes on many different populations. So of course, if we think about something like deforestation, we know that this is definitely going to impact the wildlife populations. So where these populations are moving, how close they are to households, how they're using the landscape. We've already seen some really nice examples about how this impacts the vector ecology. So kind of the mosquito development, the distribution over space and time of these mosquitoes. And finally, we know that this also impacts our space use. So if an area is being deforested, there's often someone that's moving into an area to actually conduct these activities. These people might have different levels of infection or immunity to malaria already, and different levels of health system coverage. When we kind of start to think about all of these different processes, one of the kind of key factors that emerges is we really need to think about how we understand how land use is changing and how we can monitor these changing environments. And the data sources we often use to do this are Earth observation. So this really refers to any type of data that's used to observe processes on Earth. Most of the data that we use is satellite-based. So there's a very long history of Earth observation satellites. Since over 50 years now, the Landsat satellites have been collecting data on land use, on land surfaces, land cover surfaces. But what we've seen in kind of very recent years is there's really an increase in the quantity and the quality of this types of satellite data. So we have much better temporal resolution, we have much better spatial resolution, and more different types of data that we can use. We've also seen during the same times that we have increases in new technologies. So for example, we have these drones where we can do very targeted mapping and collect much better data of specific areas that we want to map in very fine detail. So we really have kind of a new ability to understand these land use changes and map them as they're happening. So what are some of the things that we can actually see through Earth observation data? Of course, we can't actually see the parasite, so we can't see malaria transmission happening directly, but we can observe many of the environmental factors that are really impacting the transmission cycles of these different parasites. So in some cases, we can actually directly observe water bodies or breeding sites where the mosquitoes are breeding, so we can try and think about how we identify these and could target vector control there. In other areas, such as where we work in Southeast Asia, many of these breeding sites may be under trees, but we can look at how we can better understand landscape composition. So specific types of forest or canopy structures or trees where these breeding sites are likely to occur. And increasingly, we're thinking about how we can use this data to better map human and wildlife host distribution. So kind of understanding how people are using in the environment, if we're seeing signs of new logging roads or areas which are being developed, and which populations may actually be at risk. And so just to give an example of what some of this data looks like, the data and the maps at the bottom are actually data from Landsat satellite. So this is data that's collected by NASA. It's freely available to everyone in the world, or at least it was or continues to be, we'll see. But one of the kind of key things that we need to do is we need to process this data to identify specific types of land use changes or land cover that we're interested in. So the example above is showing a machine learning approach to actually classify this, to pick out areas of deforestation and how these are associated with different types of land use. So small scale agriculture, larger scale plantations, the kind of development of new logging roads into areas, or even kind of mines or other developments which may impact forest cover. As we have kind of more and more of this very high resolution data, for example, some of the data that we collect with drones, we've been thinking about how we use new types of AI tools to actually analyze this imagery to identify the types of features that we're really interested in and may impact malaria transmission. So this is actually an example using this model to pick out specific areas which we think might be likely to have breeding sites or would be water bodies which we would want to survey within this area. So we have really a lot of new kind of different types of data and new tools to analyze these data. Of course, it's not just enough to monitor the environment. We need to really understand how these different populations are interacting in these different environmental spaces and how change is impacting these different populations, driving disease transmission. So some of the ways we've been looking at this for Plasmodium knowlesi is trying to understand better how landscape actually impacts macaque home range and habitat preferences. So this is actually drone data. This is data, the kind of points or data that was collected by a GPS collar on a macaque. So you can see kind of the daytime points where the macaque is going during the day. and in blue are the places where the macaques are roosting at night. So where they're most likely to be when the mosquitoes are most likely to bite. So we can kind of better understand how these different types of land use change. So here, the edge of the plantation is actually driving macaque behavior. Of course, we can't catch all the macaques and we can't color all of them. So it's very cost effective and labor intensive to think about how we do this. So we've been thinking about what are other opportunities for new technologies that we could use to monitor some of these changing risks. So if you look on the side here, this is actually a thermal imagery. So this is some work that was done with colleagues in Malaysia. But looking at the heat signatures of monkeys. So you can see first one at ground level, very clearly there's a monkey and you can kind of see it through the trees. But if you look below, we actually put this thermal camera on a drone and flew it over areas. And if you see the kind of small hotspots, so there's some small dots within this kind of purplish area where the forest is, those are actually non-human primates roosting at night. And our field team is in that small boat in the river at the side of it. So we've been thinking about how we can kind of more rapidly use these types of technology to collect better information on wildlife population distribution. We're also increasingly thinking about what other types of sensors we could use. So the other example here is actually a small microphone. So we put the microphone in the forest, we leave it there for months at a time, and we collect very short sound snippets of different wildlife calls. So we use AI algorithms to program them, whether this is a bat call, whether this is a macaque call, whether we have examples of people moving into the forest at particular times that we're interested in. So how we can kind of better understand how these environmental factors are driving wildlife distribution and their potential interactions with people. Of course, it's kind of not just a question about where are animals, it's also where are the infected animals. We conducted, this was published in 2023, so there's certainly more work that's been done since this. But we did conduct a systematic review to understand the distribution of infected wildlife reservoirs for Plasmodium knowlesi across Southeast Asia. So we assembled all of the data we could find on any non-human primates that had been tested for Plasmodium knowlesi using molecular methods and blood sampling. So this was over 148 unique surveys that were done across this region and over 6,000 non-human primates. So if you look at the colors on this map, you can see most of these are long-tailed macaques, and there's some samples of pig-tailed macaques. Very few other non-human primate species have actually been tested within this region. And really, a lot of the work and a lot of the data has come from Malaysia. Putting all of this together, I think one thing that quite surprised us is that there's a lot of heterogeneity in where macaques are infected. So if we look at Borneo, which reports the highest number of human cases, this also reports the highest infection prevalence in macaques. And we kind of see very kind of distinct spatial patterns across different areas, but with also quite high uncertainty. One of the things that we found is that when we look at finer scale of some of these patterns, these were really strongly associated with forest fragmentation. So areas with these very patchy landscapes had some of the highest prevalence of macaques. And of course, if you look at Thailand, when it's time when we did this study, we didn't have any records, but I think this has now changed quite a bit. So interpret with, yeah, I think maybe needs to be updated as well. But we do kind of continuously find that long-tailed macaques have the highest prevalence as well. So environment is really driving infection within the wildlife reservoir, as well as kind of this reported cases in people. As you know, of course, we're looking at macaques, we're looking at landscape. We also need to look at mosquitoes. So trying to understand how these different habitats impact the distribution of mosquitoes within these landscapes. So we've done a lot of different surveys, looking at different types of forest, different types of agriculture near houses, conducting human landing catches, so catching mosquitoes outside at night to kind of better understand the number of mosquitoes, of the Anopheles mosquitoes that might be coming to bite people, but also increasingly thinking about how do we understand the mosquitoes that are biting wildlife at the kind of canopy tops or kind of tops of trees. So just to summarize a bit of this data, because this is work that's gone on for many, many years, But one of the key things that we found looking in Sabah, Malaysia, is that our predominant vector is Anopheles bilobacensis. In contrast to some of the other areas, we see that this is really biting early evening, so between 6 p.m. and 9 or 10 p.m. So this is really when people are very unlikely to be using a bed net. You're probably out doing other activities and doing different things. And this is predominantly biting outdoors as well. We, of course, with different changes in temperature and precipitation, we do see seasonal variations. And we've used some of this data to actually make these predictive maps. So we see differences in spatial abundance as well. So some areas have very high densities of these mosquitoes, others have much lower. And finally, kind of our last piece of the puzzle is really thinking about how we put people on the map. So how do we understand human movement and behavior within these landscapes? So if we're looking at a mosquito that's biting early evening times, where are people spending their time in early evenings? And so one of the ways that we've looked at doing this is actually doing a large-scale GPS tracking study. So we're collecting a lot of data on where and when people are entering different environments, so we can kind of understand how different people or different groups are using these environments. From this, we're able to look at individual heterogeneity, so who is most likely to move farther, who is most likely to spend more time in different forest environments. But we're also able to build up these kind of predictive models of community level space use. So for a given community, how many people are likely to be outside in a specific area at a specific time. Kind of putting all this data together, this can really shed a lot of insights into understanding how landscape actually impacts Plasmodium knowlesi at risk. So we have our data on land cover. We're able to map land cover as it's kind of changing. We have these estimates of human space use, so we have an idea of who is using what areas during what time. As well, we have these broader estimates of mosquito density over space and time, and the probability of these mosquitoes actually being infectious. And what this showed us when we put this all together is this actually went a long way to answering this question about where are people exposed to zoonotic malaria. And so while this is happening in forest environments, this is also happening in areas that are really quite close to houses. So really these forest edges are one of the main drivers for where these different populations interact. Kind of moving on from this, a lot of the previous work we've been focused on is really thinking about how do we understand impacts and understand risk. But kind of moving forward, we really like to think about how do we use some of this information to better design disease surveillance and ideally to prevent risks. So we've been doing a lot of work thinking about how we can link health and environmental data. So how can we collect better data on the different types of environments where people are either living or different places where people are moving to? And can we start to think about some of these sources of satellite data where we're kind of getting these almost near real-time alerts of deforestation or other land use changes, aggregating these alerts, thinking about how we can deploy these to people working in the field, and ideally alert authorities to kind of undertake some sort of action or think about how we better design surveillance and early warning systems. Our kind of work falls under a kind of much broader body of work that's done across this region. So we have been leading a climate and environmental change interest group for the Asia Pacific Malaria Elimination Network. And we're really trying to think about how we bring together different groups that are trying to address these questions. So within this broader group, we've been thinking about what are some of the impacts of climate and environmental changes on malaria in the Asia Pacific region. How can malaria programs actually adapt to a changing environment? So what are some of the practical measures we can take? And what are actually the opportunities for us to mitigate climate and environmental changes, or what is the role of the health sector in general? So we're kind of really looking forward to continuing this work and these discussions. So with that, I will just thank the very many people who have been involved in this work over many years. And thank you very much.
Thank you very much, Professor Kimberly, for your highly informative presentation. So next, we will move to the panel discussion section for today's webinar. Please.
Okay, so next will be panel discussion. So before we dive into the panel discussion, I would like to highlight our global participant we have here today. We received so many questions. Thank you for sending us many great questions. Our expert will try to answer as much as they can for today in this section. So we will go through the question. Okay, so the first question asks, which climate data source is best to use in analysis, satellites, or reanalysis in situ situations? So for these questions, maybe Professor Kimberly can answer.
Yeah, it's a really good question. I think it really depends on the purpose. So what's the kind of health data you want to integrate it with? What's the kind of role of the analysis? Are you looking to kind of predict something, or are you trying to understand some of the driving risk factors? So I think there's a lot of opportunities to integrate different sources of climate data, as well as kind of different types of satellite data or other kind of data where you can monitor environmental variables. But yeah, it will really kind of depend on the end use.
Thank you, Professor Kimberly. So we'll move to the next question. The question asks, why do two villages located close together have vastly different malaria cases? Maybe this one will be at the Jetsumon.
Yeah, from our experience that working in the village, not that far away. So what we found is that the breeding place, is a little bit huge. I mean, impact on the species composition in each area. And since the study site that I show you, all both sides have different species composition. So we found that besides the microenvironment that may impact, microenvironment, even the village is just like less than 10 kilometer apart, but the plantation around and the humidity and temperature can be much different. Okay, we suspect that, but this time we also going to monitor microenvironment in each village that we collect the vector. We hope that that can confirm our hypothesis, but for sure the species composition is different, although some major vector like minimus we found everywhere. So I think minimus can contribute at certain level, but we also found secondary vectors many all year round. In our study site, the peak transmission of malaria, generally we can see two peaks, but with those two peaks beside minimus is the majority. We also have different group of vector, secondary vector that pop up. So for sure, the vector species, secondary thing that we haven't done much work, but should be done more is the human behavior, the risk behavior. We haven't explored in detail for the past two phases, but we plan to do that for this time. So I hope that we get clear answer.
Thank you, Ajahn. So it's depend on the vector locations and the human behavior. So it's that's what make the different malaria cases in different area. Okay, so for the next question, how can we attribute climate change to changes in malaria transmissions? In essence, how should we handle confounding factors? This one, maybe Professor Kimberly can answer.
I can try. I think it's a really important question to ask, and it's it is really kind of key to think about what are we actually measuring and what are we adjusting for. When we're looking at climate change, we're really looking at long-term, multi-decadal changes. So having the data where you can actually look at things across that time span, particularly some of the malaria data over that kind of, or health data across that period, can be really challenging. I do think, any kind of model needs to actually incorporate other confounding factors or other factors that really impact malaria transmission, such as some of the very effective malaria control measures, other factors that we've seen, kind of changing land use, increasing urbanization, changes in species. So it is really important to consider all of these factors. There are a lot of studies that are starting to look at climate change attribution to health impacts. So looking across different scenarios at what would be the counterfactual if climate change hadn't occurred. And there is kind of ways to think about how to do that kind of mathematically as well.
Thank you, Professor Kimberly. Hope this answer helped. So next question, how can we immediately control mosquito breedings? This is a question for Atente.
Yeah, I'm not, I have to declare that I'm not vector biologist, but since I just chase the vector as a host of my parasite, right? So I think for malaria, depend on where you're talking about. Okay. If you talk about Stephensii, which is the major vector in many areas in Africa now, Stephensii breed in more urban sites. So maybe you can identify the breeding place easier. There are so many biological control that you can try. Okay, I never tried with Stephensii because that's not the major vector in Thailand. But for Thailand and many areas, especially like Malaysia, you know, the breeding site is not that easy to access. Okay, so to do immediate control may not be that immediate because you have to you have to find where is it first. Control adult may be more practical, okay, in this way because the breeding size can be more than one type. To identify the breeding place is tough for many areas in Southeast Asia because the nature of the vector is more um, rural, close to the forest, okay, near the forest. Some species like in rice fields, some like vector in Korea, okay. Um, I would try immediate thing is the larvicidal bacteria, which is work well.
Thank you, Atan. So it's not easy to immediately control the breeding size. So there are so many factors that you have to consider of. Okay, so moving to next question. How do you tackle the problem of estimating mosquito abundance with limited data? So maybe Professor Kimberly can answer this one.
Yeah, I mean, it is always really a challenge. So I guess one thing is, I think the data on the ground is really important. So, whatever model you fit, you do need those ground observations and you do need some kind of data that's kind of really quite critical. The way that we've approached it is we've been looking at trying to identify different types of environmental predictors that are good predictors of mosquito abundance and building up some of these geostatistical models or different types of models that incorporate both kind of space and time and different environmental predictors that are associated with mosquito abundance in specific settings. But I guess is kind of Jetsumon has already said, it really depends on the mosquito. So what you're looking for, how predictable it is, and whether you're able to kind of reliably identify these.
Okay, thank you for this question. Next question is questions related to PM 2.5. So relationship between PM 2.5 and malaria. Maybe Jetsumon can answer this one.
It's very interesting. The question doesn't say that malaria is mean for human and for mosquito, right? If you talk about the malaria patient, maybe impact for sure because the patient's already very weak. PM 2.5, we know that is also interfere because it's very small, depend on where it go, right, in the body. But if you're talking about the mosquito, it's also depend on which vector you're talking about. Like I said, if the vector is more like forest type, uh, I don't think we have any issue of PM 2.5 in the forest, right? So you're talking about the urban malaria, like Stephensii or something like that. I'm not so sure how well it's already adapt to the polluted environment. And then the PM 2.5 is so small. But we had a talk about this question this morning. One of my faculty medical doctor is interested in the question of how PM 2.5 impact dengue transmission in Bangkok. And he did a small study and found that if you flow the PM 2.5 particle into the mosquito cage, which is Aedes aegypti, but that's not malaria, like that dengue and more happen in urban situation like in Bangkok, in big city. So it's interesting that the data show that the mosquito die because of the PM 2.5 significantly compared with the normal air. Okay, so maybe that's something that PM 2.5 impact, but for sure, even we know it may kill the mosquito, be careful, it's also impact the human. So this cannot be the intervention.
Thank you for the question. It's very interesting Well, the mosquitoes, it ties on the PM 2.5, so maybe there's relationship between other dengue infections, maybe not malaria. Okay, so we'll move to next set of questions. So the first one said, what are the differences in the relationship between malaria climate and malaria transmission in Asia versus Africa? Maybe I can say someone can answer. Yes.
Well, my guesstimate, based on theory, would be that it depends on the vector, right? Because if the vector in Africa tolerate the wide range of the climate change, then it's no impact. You know, so we know that in Asia, like I already mentioned, the humidity, the humidity, the heat, the light, dark, all physical factor impact the vector, but that also impact the way the human behave. So it's not only the vector, it's the human. So in Africa, I'm not so sure how that changed the behavior of the risk population besides the vector. Okay, so it's hard to say. I would say that depend on the group. So you have a chance to get its increase or decrease. If you ask about Korean, I think I'm pretty much easier to predict because they're going to have a more months for transmission. So the case will be increased for sure because they have only Plasmodium vivax.
Thank you, Dan Jetsumon. Professor Kimberly, do you have anything to add on for this question?
No, I think that was a good answer. I'm laughing a bit because I think all of our answers are, it depends. But I think it is very true. It's very context specific and it's really depends on the specific vector as well as the kind of human population at risk in the health systems and other factors as well.
Okay, thank you. So we move to the next question. Is climate change a favorable or unfavorable factor for controlling Anopheles mosquito populations? Maybe, Professor Kimberly?
It depends. So, you know, certainly we expect that there'll be some areas where climate change will really increase the number of Anopheles. So you often see about high elevation areas or kind of latitudes that haven't had so many kind of Anopheles species that maybe this could kind of push them further. And there may be kind of other cases where this decreases. So when we look at Anopheles species, there's quite a kind of, you know, a specific range of temperature and rainfall conditions that are suitable for different species. So if it doesn't rain at all, you won't get Anopheles. If it rains too much, it will flood. So kind of where are these areas where you're hitting that threshold where Anopheles can fly, kind of really thrive. And it kind of that's very true with temperature as well. If it's too cold, the population won't be able to survive. And if it's really too hot, eventually the population will die. But at kind of some of these increased temperatures, we will see these kind of much faster development rates in the mosquitoes.
Adam. Okay, I'll move to the next question. What preventative measures should a country adopt to certify malaria free?
This one, I assume that what the country should concern, right, should take the effort to do to ensure that we get malaria free certification. I think one thing that we know is always helpful to control or to keep the malaria cases as a lowest is vector control. So strengthening vector control, you cannot just compromise or dilute or make it easy for vector control, especially the area that used to be the hotspot of the malaria, because it's possible that you still have a lot of asymptomatic cases walk around. So if you control the vector low enough, then the chance to re-emerging after you clear many of the symptomatic case will maintain. That's the one thing. The second thing for sure, I think many countries already adopt 137 when you have case to make sure that you clear any potential case around to ensure that a little bit like semi-active surveillance, but not doing it as a whole village all the time. So two things. Yeah, I think.
Right now we'll pause this section for a bit because there's someone in a participant. He wants to talk to the expert directly. Mohammed Andres. Yes, please. Please unmute.
Hello, everyone. Yes, I want to ask something. What is the biocontrol of malarial mosquito? Like in sugarcane, there are some biocontrol agents that we can use for their control. So for mosquito, there are some biocontrol agents are used in the world or not?
Do you catch the question? Can you repeat the question, please?
Um, Let me clear again. I want to ask what is the biocontrol agent of malaria vector means the mosquito, like in different crops. For insects, we can use biocontrol agents, means through the living organisms, we can control the pests like malaria.
So you asked about what is the biological control, or you asked if that biological control also impact for the other thing. Not so sure. You asked about the brand. That is bacteria. You know about the BTI. Okay, that already some company make the commercialized by bacterial that can control the larvae available. Okay, I'm not so sure how that continue now, but there are many try in the rice field environment or something like that. It show it work. Okay, but that one did not work well with Thailand because the breeding site is slightly different. Not the state still like rice field or other agriculture area.
Okay, thank you. I hope they answer your question. So we will continue the question from the registration. So the next question is, what is the current status of WHO supported EORs? Can the system be accessible by communities and village health volunteers?
Yeah, so I actually can't answer to what the current status is. I know it has been implemented in several countries. So the EWAR system is the early warning and response system. This is predictive models, which are using, I think, typically malaria cases or other kind of health impacts with predictors, such as some of these climatic factors. So, for example, rainfall and temperature, to forecast and give some sort of alert and when diseases are likely to occur. So I'm not so sure about in this region what the status is and whether it's accessible by different groups. There are a lot of other really kind of quite successful early warning systems which have been implemented for vector-borne diseases across Southeast Asia. So for example, DMOS, which is using satellite data and climate data to predict dengue in, I believe, Vietnam and Malaysia. So there are kind of a number of these different systems which are being developed and being used to actually give these kind of early warning signals so that there can be some sort of proactive response.
Thank you. We'll move to next question is, what collaboration goals or plan exist between the malaria control effort and the pest control industry at the grassroots level? Yeah, Jetsumon.
Yeah, I think it depends on the country. I understand that in Thailand, the ministry that take care about the pests is involved with agriculture, so not Ministry of Public Health. Okay, but on the other hand, for vector control is under the Ministry of Public Health. If they both can talk and share resource and share the budget and make it more efficient, it will be possible. But for those of you who work with the government agency, you know that how difficult bureaucracy or the barrier or all the detail. But you are right, you know, they can combine it. But one thing we found very interesting in Thailand is that we thought that we in many area that close to malaria transmission will have heavily use of chemical for pest control for that agriculture that may impact on the vector malaria vector insecticide resistance increase like they found in Africa. We found that is not the case, so not so sure how these two group can help.
Thank you. Well, we have last two questions from the registration. So next one is, what is the impact of long-lasting insecticidal net mass campaign on malaria?
If you talk about campaign, it means the campaign to encourage the villagers or the population at risk to use, how that impact, I would say that is fail. But if you're talking about if the mosquito net itself, it should be good, you know, I think it's good. The only problem that it not really impact in terms of malaria control in Thailand because the population will not use it. Okay, so if the people use it, it would be good. So that means the campaign to promote using, at least at my study site that we work, I can say that it's completely failed.
Okay, so it's really depend on the usage of people. Thank you, Dan. The last question. Okay, so for the last questions is on cost-effective solution to control malaria. So Jetsumon can share some this cost-effective solution to control malaria.
Cost-effective solution. I think the lowest cost, and it should be the most effective, is try to find the excellent way to educate the population at risk to protect themselves from mosquito bite. That's it. I think it looked like the tool to use is not that expensive, but the right way to use is very challenging. You have to talk to different population at different economic, socioeconomic background to make them understand that, you know, the key thing is that if you don't let the mosquito bite, you won't get vector-borne disease, right? And how to protect yourself from mosquito bite and why is important. Malaria is a disease that can kill you, depend on the species. No sign in the south, kill. Okay, all over Thailand now we have only Vivax that not really kill, but you can be very sick. Okay, you cannot work. It also can impact the young kid, something like that. But how to protect? That's the best way because as if you attend from the beginning until now, you know that we have so many species that you have to deal with different things. The climate keep changing, the people keep moving, so it will not be one size fit all unless the malaria risk population understand this and try to protect themselves. Right now, the government provide the bed net, they don't use. Okay, the government try to spray the house. Some of the house owner don't like it, not allowed. Okay, and when they get sick, especially malaria, we treat them. Unfortunately, for Vivax, you still have to take 14-day primaquine unless you confirm with the GCPD, then you can take one dose tefenoquine. But with that, if they don't comply, they don't complete that, the malaria still continue. Okay, so how you make everyone aware of this and don't let mosquito bite you, then you won't get malaria. I said, I think this is the most cost effective if you find the right way to do.
Thank you, Ajahn. Yes, the most cost-effective solution is just don't let mosquito bite you. Okay. Now we'll move on to the questions Q&A from the Zoom webinar. So Wisawa asks, what are the main challenges in addressing climate change and malaria? You both can answer. So, what are the main challenges in addressing climate change and malaria? This is from the Q&A from the audience.
It's all climate change. I think there's there's quite a few. So one of them is just having the data. So we're able to identify what are the impacts and where are the impacts and what are the regions most at risk. I think one of the things that we've been thinking about through APMEN and through other groups are what are some of the effective adaptation strategies? So how can we kind of use what we know about impacts to look at planning? And I think this is still a real gap. So we've seen a lot of these early warning systems, which are really useful for thinking about planning on kind of a short-term basis or potentially alerting when there's kind of times where we might have increased malaria risk. But we also kind of need to think more broadly about how do we kind of think about addressing disasters or other situations. How can we kind of have more of a response where we're prepared for some of these really extreme weather events and their impacts? So, anything you wanted to add? No.
Okay, thank you for the answer. And next, we have an interesting question from Muhammad. He asks, what do you say about the control of female mosquito through pheromone traps? Because male mosquito is harmless.
Well, I'm not insect biologist, but I would say that the pheromone is very specific, right? And if you have so many species, so you're going to have a cocktail of pheromone. Can you do that for the trap? Because we found that many groups try to identify the odors that track many species of female to make the trap, to collect these female. Problem is that you can do as much as evaluation in the lab with certain species that you can colonize in the lab. But then when you bring these to the field, it's a completely fail because the vector species is not the same. The environment in the field is not like you control in the lab, right? The wind flow and what attracts. So we found that this trap, we tried one trap with the scientists from US designed by identify the chemical that really attract the female the most, use the 3 species, the lab strain. And when they make the trap and we try at our study site, it completely fails. CDC light trap, you still get the most of them. Okay, so theoretically, yes, how to evaluate that and which species you evaluate that, not that easy. But I don't say that anyone that work on this should stop, but I think you have to find the approach that can apply to majority of the species. Okay, and which go back to, do we know which area has which species of the vector in that area? Even in Malaysia, there has a lot of the study on vector Fornace. I think they still have a lot of mystery to identify. Okay, in Thailand, we don't know which species yet, but whatever they said happened in Malaysia, all of those species, we have it all in Thailand. But whether all of them are the vector, we don't know.
Okay, thank you, Dan. So we have next question from Siri Pin. She asks, climate change is closely linked to natural disaster, which poses challenges to the accessibility of health service and increase the risk of disease outbreak. In your opinion, how can we utilize data for policy development and effective response to these situations? Maybe Kimberly can answer.
It's an excellent question because I think many people that work in the region in this area know that some of the most severe impacts that we've seen on malaria have been in response to these extreme weather events. So in particular, when we think about Pakistan and the 2022 floods, when the cases kind of went from a few 100,000 to millions every year. And this was largely due to displacement of people. Obviously, if people are displaced, vector control can't be deployed. There was also a change in the vector species. So there were a lot of factors that contributed to this kind of changing ecology. So I think it's one we've been thinking about a lot, and I think there's kind of a lot of lessons to learn from places that it has happened and how we kind of respond to it. It's certainly when we're looking at these kind of very extreme events, they're very difficult to predict. So it's hard to think of having an early warning system where we could predict a natural disaster in the time that it would take to prepare. But I think there are really kind of key lessons that we can learn from other experiences of these. And we can think a lot about how we use some of the kind of data and the model predictions to identify areas where these are most likely to occur, areas where we think these are most vulnerable, and where there might also be malaria transmission or transmission of other diseases.
Thank you. Thank you, Kimberly. So we have another question from anonymous attendees. The question is, please, I need clarification on this phenomenon. I don't find the malaria vector in hyper-endemic environment, but people are treating. What is the implication for the disease transmissions? Is it that the Achilles vector found on the carrier, or the very few are transmitting it? Thank you. Jetsumon, do you understand the question, or you want me to?
Not so sure I understand the question. What I mean, hyperendemic environment. What is a hyperendemic? Yeah.
Okay.
But for sure, QLEX is not the vector.
Curious. Yeah.
QLEX is a vector of reptile malaria, not human. Yeah. I think it's reptile. It's avian. Yeah.
Okay, we can move to next question. And Miss, in a resource limitation, how can we maximize malaria control effort while addressing the challenges posed by climate change?
I think the vector control measure will be the easiest one. You know, the people get used to, they have the bed net provided, use it. Okay, and the government can still do at least minimum vector control, especially in Thailand, 2 times a year. You know, like active doing that. Besides that, I think, except it's flat, that go to back to Kim answer about Pakistan issue, right? But in general, if it warmer, longer time for transmission or something like that, so I still, at least, you know, that the two things that I think we must do is a vector control, and I think surveillance Thailand is very good already. So the vector control and self-protection by the population at risk.
Okay, thank you for the answer. We have some questions from our audience in the room. Maybe Soda Mini.
Thank you so much for the presentations, really insightful and eye-opening for those of us who are not that familiar and new to the field. So maybe they may be slightly basic questions, but Dr. Kimberly, you mentioned about the use of AI in your data collection efforts. Could you share a little bit more about how this is being used and how it increases the accuracy of the data collected or interpretation? And what would be potentially next steps going forward? And if I may just follow on to Ajahn Jetsumon as well, and perhaps you too, Dr. Kimberly, you highlighted the importance of multi-sectoral collaboration. A big emphasis of your work, both of yours in some ways, is on strengthening data collection and surveillance systems. How do you see researchers in, say, the social sciences? You mentioned behavioral interventions are the way forward. How do you see other groups utilizing this in a more effective way in Thailand or even at the regional level? Thank you.
Excellent. Well, I can take the one on AI first. What we've noticed is, so kind of some of we use AI in different ways, but really the main way is to classify either environmental data or some of the other data types to pick out specific signals. So for example, AI has really revolutionized our ability to kind of rapidly identify from imagery. If we're looking for example, where are the houses? Is this a specific forest type? Trying to segment and identify that image. So that's really increased the speed, and it's also made it much more feasible to do with some of the very, very high resolution imagery we have. So if you think of like the way your phone works with photos and it can kind of match them into different groups, actually we've seen that those algorithms have gotten a lot better as we have more and more training data. And we do something quite similar for sound data or for other data where we're trying to identify animals. So we don't want to listen to it the whole time. So we could kind of actually pick out what are the signals that are likely to be different animals. So I think there's a lot of capacity. And as we have these absolutely enormous data sets, even just managing that data, AI is a really powerful tool. It does have its limitations. We still need the training data. We still need to be confident of what we're teaching AI to find. But there is a lot of potential.
Yes, I think you asked who should be the one that use all of those information. I think anyone that involved with the especially for malaria, it should be the policy maker or strategic planning at the regional level, like Office of Disease Control that we have in different sites in Thailand, or even the central MOS should understand and try to utilize those information. That would be helpful because different group will collect the data in under different expertise that they can, right? But I think the challenge is that how you combine those information and turn it to be best practice policy that can implement in many sites. Even Thailand, not a big country, right? But it's not one size fit all for all malaria endemic, and we saw that. Okay, by the time that many sites, the malaria cases decline almost zero. One thing that gone before malaria is the expertise in that area to do case control. And then once you don't have anyone understand how to make even better control or decrease the case come back. And you don't have anyone to realize that, oh, it's come back, until it's become a very big problem that even non-malaria expert can pick up. And then you go back to the starting point again, try to eliminate malaria in those areas. Although the country did well, that's why we reduced from more than 1 million cases when I start working in malaria to less than 20,000 cases now. But the hotspot still be hotspot. We still have hotspot.
Okay, so there are no more questions, we'll move on to the next session. So we're done with the questions for this webinar. Thank you so much, everyone, for asking questions. So I'll move to the next one. So I will just briefly summarize what we learned from today's webinar. So here are the key takeaway. So we learned that malaria is a climate-sensitive disease. With increase in temperature, humidity, rainfall, and drought, it's impact the mosquito breeding and also increases the malaria transmission. And also deforestation bring mosquito animals and human closer together, so it increases the risk of infection. And there are also a lot of increased cases of malaria in Thailand, especially in Thailand-Myanmar border due to the migration, weak vector controls, and climate change. And also we learned about the mosquito behavior. The biting patterns are very important as well in controlling the infection. And with all that said, better surveillance is needed. So using satellite data, AI, and predictive model, they can help and help track and control the malaria risks. Okay, maybe like one more question from the anonymous attendees. Said, can you share the resources where we can get the most reliable weather/climate data for free?
I guess that's me. There's a lot of different sources. So again, it kind of depends on your application. So most countries have their own Met services and have ground stations and access to different data sets. You could also look at some of the reanalysis data sets. So for example, Copernicus, which has an online platform where you can log in and actually download data. So like the ERA 5 land data, which has temperature, humidity, other variables that are important. But I think with all of them, there's kind of almost like an infinite amount of data. So it's just really kind of important to make sure that it has the temporal and the spatial resolution you need, and to be kind of clear about what are some of the kind of issues with accuracy and data quality in those different data sets. Yeah.
The Brazil one. Okay.
Okay. Given the current trends in climate change, what are the currently feasible environmental and climate strategies that can be implemented? So this is someone from our group. I recognize the name. But it's a good question. One of the things we're really interested in, I mean, I think we certainly need to think about how we use data to better identify the areas and the times where we're most at risk. So I think that's a kind of a key intervention, and surveillance itself is a key intervention for malaria. But we're also really interested in thinking about what are some of the ways that if we know that certain types of land use are associated with increased risk, can we design land management strategies that reduce these risks? So what are the kind of opportunities to actually decrease some of the risks that are kind of driven by human environmental changes? And alongside of that, if we think about different interventions which are being undertaken for climate change, so for example, reforestation, how we can, what are the impacts of that on malaria risks? How can we monitor them? And how can we kind of make sure that we're implementing them in a way that ideally kind of supports health as well as other goals? So there's really like quite a long history in malaria of using environmental management to reduce risk. But it's kind of making sure that each solution fits the context and fits the environment.
I think the last one is Brazil, USA. I'm not a fan of genetic modified malaria vector. Especially, we know that in Asia there are so many species. So you can do only a few species. You can try to do sterile male of a few species, but not all. And at the end, we did a study briefly on the vector community. When you get rid of one vector species, the other grow up. So it doesn't help until you have to do for all. In one of my old study, one breeding place all year round, we collect more than 20 species of Anopheles in a small breeding place. And this population just keep it growth, okay, all year round. So let's say if it's just one small village with 700 people that the malaria transmission heavily, what are you going to do with this technique? I don't see that you can do all sterile 20 species, right? So it's not that simple. In Brazil, maybe they have less species, or they identify the major vector. In US, I'm not so sure with species. I'm not familiar with this word, but interesting approach. A lot of people try to do a genetic modify, like in Africa, they can do that because the major species that they found is feasible to do that technique, but I don't think it's feasible to do that in Asian mosquito.
Okay. Okay, thank you for all the questions, and also thank you for answering the questions. So this is the end of the webinar. Thank you everyone for joining. We hope it has been very informative for everyone. So this is our first webinar. So please keep an eye out for this episode 2 of our webinar series. Thank you, Professor Kimberly and Professor Jetsumon for joining and giving us special talk for this section. Thank you very much.